Félix Balazard, talking about AI for evidence-based covariate adjustment in clinical trials. So first, I'll talk about the concept of covariate adjustment in clinical trials. Then I'll discuss what are the determinants of power gains from covariate adjustment, how covariate adjustment can help broaden the eligibility criteria of clinical trials, and the opportunity that is represented by the new covariates using deep learning on histological slides through covariate adjustment. So first of all, what's covariate adjustment? So covariate adjustment can increase the statistical power of randomized clinical trials. So first of all, what's a randomized controlled trial? You're basically testing the presence of a treatment effect. So you want to do a statistical test whether B1, a coefficient for the treatment allocation, is equal to 0 or not. And you have a clinical outcome, Y, that you want to improve with your drug, and a treatment allocation, T, that's going to be randomized. And then you can have some information about your patients. Here we'll say that we have a prognostic covariate X. So a covariate X that is associated with the outcome Y. And so what does, how can we take into account this prognostic covariate X? So if you do the standard, the simple unadjusted analysis, you're simply going to look at the outcome in the two arms of the trial and try to find a difference in means between those two distributions. And when you do the adjusted analysis, you're basically going to add B2X here in this regression model, and this will basically mean that you're going to look at differences in mean between Y minus B2X. So once you removed B2X from the outcome, you're going to get more peaked distributions because you've removed some of the variation in the outcome that is explained by the prognostic covariate. And because you have more peak distributions, you will gain statistical power, and you will have a more precise estimation of the treatment effect B1. So how can we understand this in a more simple terms? So it's really about signal and noise. So the signal you care about in a clinical trial is the treatment effect, and the noise is everything else, such as the variation in outcome that is captured by prognostic covariates. And so we can think about signal and noise in a sound way. So if you're listening to music on the subway, the signal you care about is the music that you're trying to listen to. And you have all the subway background noise that's going to cancel out your music and stop you from enjoying the music. And so you can increase the music volume. So in the clinical trial, that would be increasing the sample size. Or you can have a noise cancelling headset that's going to actively cancel the noise around you, the background noise. And that would be the equivalent in the clinical trial would be adjusting on prognostic covariate. So removing the noise that is stopping you from listening to the treatment effects. And so covariate adjustment is not something new. We propose to do it in a more evidence-based way than what was done before, maybe. And so what does that mean? That means at the clinical trial design stage, using external data that can be past historical clinical trials, real-world data, in order to really look and train models that capture all the prognostic information that we can in the trial population of interest. So that means having training data, and we can think obviously of standard like biological variables that are usually captured in clinical trials, but we can also look at what kind of prognostic signals there is in other modalities, such as radiological images or even histopathology images. And from this training data, then we can train models, and then we can apply those models, those prognostic models that predict the clinical outcome. We can apply them in the trial using the same modality, and then adjust this, do the adjusted analysis that's going to be more statistically powerful. And so this is a case study of a project we did for a pharma client. And basically they had a failed trial where they tested their new drug against the standard of care in an advanced cancer setting. And we had a clinical trial on the same indication. And so we trained prognostic models on that external data set. And then we applied the models to the client's trial. And basically what happened, so here you see the original stratified analysis that they did. So that had a p-value of 0.08, so that's always sad to hear. And once we adjusted the analysis, we actually went below the significance threshold for that trial. And so what that means is that if they had done their covariate adjustment in an evidence-based manner, then they would have been able to demonstrate that the treatment is actually efficacious. So obviously this is a post hoc analysis, so not very convincing for the FDA. But yeah, this is the first message. Covariate adjustment can de-risk and accelerate clinical trials. So now, it's true in general that it improves power, but how much and what determines how much power is gained by covariate adjustment. So in order to understand this, we did a simulation framework using many different parameters. And the two main parameters of interest are the C-index of the prognostic covariate. So that's a metric that captures how prognostic the covariate is. So it's between 0.5 and 1, and 0.5 is random, 1 is completely determined by the covariates, the outcome. And then the other key parameter of interest, and this is actually a bit surprising, it's not previously reported in the literature, the other key parameter is lambda, the cumulative incidence of the event in the placebo arm at the end of the trial. So if you look here, that's here you have lambda equals 40%, and so that's that's a key parameter, and it's basically tells you how much how aggressive the disease is, and also takes into account the time the follow-up time. So we simulated the data. Then we had those simulated clinical trials where we could do both an adjusted and an unadjusted analysis. And then we had power curves depending on sample size. This is power, this is sample size. So here is the unadjusted power curve. Here is the more powerful adjusted power curve. And we looked, in order to quantify the gain in statistical power, we looked at the reduction in sample size for a statistical power of 80%. And so these are the results. And as I was telling you, the two parameters of interest are the C-index and cumulative incidence. And so the reduction in sample size increases both with C-index and with cumulative incidence lambda. And so you can see here, we have the reduction in sample size, the C-index, and the colors, the color code codes for the cumulative incidence. So here you have lambda equals 10%, here lambda equals 90%. So you really get a very strong gradient on lambda. And so if you have if you're looking at a pancreatic cancer, for example, where you're at 90% after 3 years, even if you have only 65, a C-index of 65, you're already going to be able to reduce the sample size by 25% thanks to covariate adjustment So it's a very, very strong gradient. So what's the key message here? So the impact of covariate adjustment is larger when we identify all possible sources of prognostic signal, so that if we maximize the C-index. And it's also more important for diseases with large proportion of events in trials. So it's going to have a larger impact in metastatic cancer. It's going to be much more impactful when if you think about secondary cardiovascular disease, where you're about 10% at the end of your three-year follow-up. It's not going to be as impactful. You're really going to need a lot of prognostic signal if you want to have something impactful for that kind of disease setting. So now we're going to try and see the link between covariate adjustment and eligibility criteria. So there is a discussion around eligibility criteria that centers around the idea that maybe we've been too restrictive in the eligibility criteria in clinical trials. We've made it very hard for patients to get into clinical trials, and that has that's a problem for different reasons. But one of them is it's getting hard to recruit patients in the trials, and some trials fail because they can't enroll enough patients. There's also a question of generalizability. Are we really sure that our treatments that we've tested in a very selected population, is that treatment going to be really as efficient in in a more broad population? And there's been this really great paper published in Nature by Genentech researchers using Flatiron Health data, so real-world data in oncology in the US, on non-small cell lung cancer trials, where they've emulated non-small cell lung cancers using real-world data to try and look at the impact of removing certain inclusion criteria, both on the number of patients that are eligible. So you see here the variables that have the most impact on reducing the pool of patients that are eligible. So for example, so it's ECOG and histology. And also looking at the impact on the coefficient of the treatment effect. And basically they showed that a lot of the inclusion criteria are actually not useful, and reduce a lot the population size, the enrollable population. And here, what we've done is look at what happens if you have, by playing both with inclusion and with adjustment. And so basically, if you look at the null model, unadjusted model, and we're going to look at different sets of inclusion on a prognostic covariate. And basically, you get, by going from the full inclusion to the restricted inclusion, you get a gain in power, simply because the population is more homogeneous, so it's easier to see the treatment. But then, if we do a covariate adjustment in either case, we get the same power curve, and that means that basically, if we have broader eligibility criteria, we're going to have more heterogeneity in the trial, but we can remove that heterogeneity analytically by doing covariate adjustment in an optimized way. And so that means that by doing evidence-based covariate adjustment, we can allow for broader eligibility criteria, and that means faster enrollment and better generalizability. And so that's another key message. Better covariate adjustment allows for more inclusive trials. And now I'm going to talk to you about things that we're really excited at Okin about, and that's new covariates for adjustment using deep learning on histological slides. So what's histopathology? This is you get cancer, you get the surgery, and then we're going to look at the tissue under a microscope, and we're going to have very high definition images. So it's really a ideal playground for deep learning. And you can see cancer, you can see the tumor microenvironment, a lot of information. And before it was, it's still looked at by expert histopathologist, but it's really ideal to automate and get more exhaustive exploration of the information that's in those slides. And so we did a project with APHP, the Parisian hospitals, where we trained a deep learning model to predict overall survival in resected hepatocellular carcinoma. And so there is currently no adjuvant treatment for resected hepatocellular carcinoma, and it's still there's quite an unmet medical need, as the death rate at 5 years is 32%. And so we trained our data, so actually on those 94 patients, and then we applied the model on those 300 patients from the TCGA. So we have a deep learning methodology that's tailored to histopathology. So it's very different problem compared to a lot of things that we see in computer vision. We have very large images with information that's very local in the whole slide image. So the tumor is going to be typically a small part of the tissue that is on the slide. And so it needs a specific deep learning methodology tailored to histopathology. So we tile the slide into small pieces, tiles that we, yeah. And then from the tiles we extract features using standard deep learning, pre-trained deep learning networks. And then we're going to do to basically score the tiles by saying, is this a tile of a good prognosis or a bad prognosis? And we're going to base the final prediction on the worst prognostic tiles and the best prognostic style. And so that's Chowder, and we have a variant here, Schmarder, that has the same, here the same thing as Chowder, with the idea is that we're in the end going to use only a few tiles because the information is localized, so we want to focus on where the information is. And also another branch of the deep learning network that has an attention mechanism that can take into account the annotations on the slide, if we have annotations. And here you see that we get quite an improvement in C-index. We're at a C-index of 0.70 compared with 0.63 for a composite score of clinical information. And then we can look at what happens if we do the adjustment on standard variables that would usually have been used, and standard the same variables plus our deep learning model, HCCNet. And so we get again those power curves where we have around 5% here of additional power, and that would have that's basically similar to having the same statistical power for 10% less patients. And we can again look at what happens if we consider different inclusion criteria. So if we take the union of all exclusion criteria in clinical trials, we get only 55% of patients, and yeah, the same message than before applies. We can have broader eligibility criteria when we do covariate adjustment in an optimized way. And yeah, this is not new for the regulators. They've published guidance on the subject, even as recently as last year for the FDA draft guidance. We've discussed about the EMA, about the relevance of adjusting on deep learning histological covariates. They had positive feedback, still some work to do, but overall positive. And yeah, that's my final message. Prognostic signal in new modalities unlocked by AI can impact clinical trials through covariate adjustments. Thank you for your attention, and feel free to come talk to us.